Plants are living. They need food and water just like human does. In our human body, we have circulation system to transport and translocate water and nutrients to the organs and the cells. Plants almost do the same thing. Long-distance transport systems are integral to successful functioning of land plants. Vascular plants are characterized by vascular systems composed of two specialized tissues, xylem and phloem. Xylem and phloem occur together and extend throughout the entire plant body. Xylem transports water and soil-borne nutrients from roots to transpiring leaves, where photosynthesis forms organic carbon compounds. Phloem transports water, mineral elements, amino nitrogen compounds, and photosynthase from fully expanded leaves or storage organs to meet the nutrient requirements of growth or storage organs. So, how do xylem and phloem look like? Xylem tissue has truncated elements, vessels, fibers, and parenchyma. It is worthy to mention that xylem cell wall is lignified to increase the strength of it. Here you can see a beautiful xylem tissue slide. A red stain shows the cell wall organization of xylem. Xylem cell is dead when it gets mature. Phloem tissue has sieve tube elements, companion cells, parenchyma, and fibers. Sieve tube element is almost empty, but it has the nucleus in it. As a hardworking plant biology 112 student and a future scientist from UC Davis, you must be curious about how this vascular system forms. Now I'm going to unravel this puzzle. As you can see, here we have a tiny cute plant. The top part of it is the apical meristem. Then the apical meristem will differentiate into three primary meristems: protoderm, ground meristem, and procambium. As cells grow and get mature, the procambium will produce primary phloem inside and primary xylem outside, and form this organized ring of vascular bundle. This is called the primary growth, which increases in the height of plants. Plants also have secondary growth. In order to grow in girth, the residual procambium will differentiate into the vascular cambium. This vascular cambium also forms xylem inside and phloem outside to enlarge the vascular bundle, and the cortical parenchyma cells will differentiate into the intervascular cambium. Vesicular and intervascular cambium join together, and ultimately a complete ring of vascular cambium around the stem leads to a cylinder of secondary growth in the stem. Ta-da! Here we have a complete vascular cambium.
To survive in various environments, plants have evolved to be made up of flexible vascular tissue that carries significant biological functions that without it, their growth would be restricted to that of non-vascular plants. In order to grow larger, plants must have a system that allows the long-distance transportation of nutrients and water from the roots of the plant deep within the soil all the way to the shoot and leaves. Plants are able to do this due to the xylem, which are hollowed-out cells. The xylem creates a positive pressure on the column via transpiration. This process occurs by pulling the water in an upward direction as water evaporates in this open stomata with the help of adhesion and cohesion forces. Additionally, the xylem consists of xylem fiber cells that have thickened secondary cell walls that provide mechanical support to the plant body. The xylem can further differentiate to create metaxylem vessels. These vessels have a net-like pattern which provides rigidity to the cells. The phloem plays an equally important role in the plant by transporting sugars created from photosynthesis in the leaves. These sugars are transported in a bidirectional flow to the rest of the plant. To do this, the phloem works in tangent to the xylem by exchanging water with the xylem, and with the help of specialized proteins, the sugars are able to travel into the rest of the plant.
How does a plant decide which cells become vascular tissue? There are many signaling pathways, proteins, hormones, and transcriptional factors involved in the development of vascular tissue. At the start of the early globular stage in the embryo, asymmetric cell division leads to the differentiation of the provascular cell layer. The provascular cell layer consists of 4 cells that are found in the center of the embryo. During the late globular stage, 4 vascular initial cells are formed. These 4 initial cells give rise to vascular cells. Different plant species have different arrangements of vasculature. The arrangement of xylem and phloem is regulated by auxin and cytokinin. Cytokinin promotes the distribution of pin 1 and pin 7, which channel auxin towards the xylem axis. The feedback loop between cytokinin and auxin controls the organization of the vascular cylinder. C3-HDZ transcription factors in cytokinin levels regulate differentiation of cells into protoxylem or metaxylem. TDIF, or tracheary element differentiation inhibitory factor, is an amino acid peptide that works in the signaling pathway with PXY. A TDIF receptor, or phloem intercalated with xylem, to maintain procambial and cambial cells, so that these cells can form vascular tissue. The maintenance of the procambial and cambial cells, which are often referred to as the vascular stem cells, allow for indeterminate plant growth. WOX4, a Wuschel-related gene, promotes proliferation of procambial and cambial cells in the TDIF/PXY signaling pathway. Glycogen synthase kinase 3 proteins, GSK3s, act downstream of the TDIF/PXY signaling pathway. The TDIF-GSK3 pathway inhibits cells from differentiating into phloem cells, while the TDIF-WOX2 signaling pathway inhibits vascular stem cells from differentiating into xylem cells. Brasinosteroids, or BR, are hormones that play an essential role in xylem differentiation. The BRL genes, 1 to 3, are predominantly expressed in the vascular tissues. BRL1 and BRL3 encodes functional BR receptors. BRL1 plays a role of BRs in xylem differentiation. BRL3 may repress phloem differentiation. It is proposed that BRL1 and BRL3 function in provascular differentiation to maintain proper xylem to phloem ratios. A synergistic interaction of BRL1 and BRL3 with BRI1 was required to achieve normal vascular differentiation in the plant shoot. It also contributes to the collateral organization of the vascular bundles in the plant shoot. The BRL2 protein has been proposed to regulate provascular cell specification. Brasinosteroids contribute to the establishment of the collateral xylem and phloem patterns. As you can see, there are many signaling pathways that determine the fate of vascular tissues. These are just a few examples of important regulators in vascular tissue development. The process is not yet fully understood, and there is a lot more to learn.